Hello everyone. Today we are diving into the world of gene regulation. Gene regulation is the control of gene expression. It is the mechanism by which a cell turns gene on or off depending on the environmental condition, stage of development and type of cell. This helps in saving energy and ensuring proteins are made only when needed. In prokaryotes, this regulation is well explained by Operon model, proposed by Jacob and Monod in 1961. They studied gene regulation in E. coli, especially how it breaks down lactose when glucose is not available. This system is called Lac Operon. An operon is a group of related genes that work together under a common control system. The Lac Operon has following main components. First, regulator genes produce a protein called repressor. The repressor controls whether the operon is on or off. Second, promoter genes. This is the binding site for RNA polymerase to start transcription. Third, operator genes act like an on-off switch. The repressor protein can bind here to block transcription. Fourth, structural genes. These genes actually code for enzymes involved in lactose metabolism. For example, beta-galactosidase, permease, and transacetylase. Let's understand how gene switching works using repressor, inducer, and co-repressor. The repressor proteins produced by the regulator genes, inactive on its own, becomes active only when bound to a co-repressor. Once active, the repressor binds to the operator, blocking the RNA polymerase, means gene off. Co-repressor, a molecule that activates the repressor, help shut the gene down when the product is already available. Inducer, a molecule like lactose, it can bind to the repressor and inactivate it. This removes the repressor from the operator, allowing RNA polymerase to function, means gene on. Here, how E. coli uses the Lac Operon. Condition first, no lactose. The repressor is active. It binds to the operator. RNA polymerase is blocked. No enzyme is produced. Condition second, lactose present. Lactose acts as an inducer. It binds to the repressor, making it inactive. RNA polymerase moves freely and the enzymes are produced to digest lactose. This system allows the cell to respond quickly and efficiently to the environment. Why is gene regulation important? Because it prevents wasteful energy usage. It allows adaptation to changing environment, enable differentiation and development in multicellular organisms. In prokaryotes, it ensures they only make enzymes when they are actually needed. In eukaryotes, the process is even more complex, but the Operon model gives us a strong foundation. The genetic code was cracked in 1961 by two scientists, Marshall Nirenberg and Henrik Mattei, using synthetic RNA in a test tube. They decoded the first codon UUU, which was found to code for amino acid phenylalanine. This opened the door to understanding how genes are translated into proteins. Let's look at the basic unit of the genetic code, the codon. A codon is a triplet of nitrogenous bases. Each codon codes for one amino acid. Since there are 4 bases and codons are triplet, we get 4 multiply 4 multiply with 4. We get 64 codons. 61 codons code for 20 amino acids. 3 codons are stop codons: UAA, UAG, and UGA. They signal the end of protein synthesis. To prove this, many scientists came together. Hargobind Khurana developed a method to synthesize RNA with specific base sequence. Marshall Nirenberg created a cell-free system to test protein synthesis. Now let's explore the important features of genetic code that you must understand for exams and concept clarity. First, triplet nature. Each codon is made 3 nucleotides. So, 1 amino acid equals 3 bases. For example, AUG, codon, methionine, also a start codon. Second, degenerate code. Some amino acids are coded by more than one codon. For example, leucine is coded by 6 different codons: CUU, CUA, etc. Non-ambiguous. Each codon codes for only one amino acid. No confusion. For example, UCZ will always code for serine, not any other amino acid. 4. Non-overlapping. Each nucleotide is part of only one codon. Codons are read one after another without sharing bases. 5th, commaless. The genetic code has no punctuation marks. Once translation begins, codons are read continuously without gap. 6th, universal. The genetic code is nearly identical in all living organisms, from bacteria to humans. For example, AUG codes for methionine in both E. coli and human beings. But some exceptions exist in mitochondria or certain protozoa. 7th, linear. Codons are read in a straight line from 5' to 3' direction of the mRNA. No loops, no branches, just a clean one-way reading of codon. Why is all this important? The genetic code is a bridge between genes and proteins. It ensures that proteins are made accurately and efficiently. It allows scientists to predict proteins from DNA sequence. It helps in genetic engineering and biotechnology. Without the genetic code, life would not be able to exist in such a coded, precise, and programmable way.